Thank you to everyone who has attended the webinar today. I'm going to talk first briefly about mite diversity in general, and then I'll switch into why ABMI monitors mites and talk a bit about how we've been using that data, and then some gaps that we might, or future opportunities that we might want to consider. So most of us are well aware of the mites that cause us problems, such as the house dust mites, which are actually quite pretty in this photograph, or ticks, which are actually mites. The mites that cause scabies or mange, or those that are, cause problems with our gardens, such as spider mites. But I know for myself, I really didn't understand the diversity of mites until I started working with them. And we find them everywhere. They can be found from the Antarctic to the Arctic. They're from the highest mountaintops into the deepest, the ocean. So we find them on the ocean floor. And again, they're both terrestrial and freshwater and aquatic. And they have all sorts, they're in all different habitats. We even find them on ourselves and on animals. They have many different adaptations and all sorts of colors and shapes. And so, for example, some of them don't even look at like mites, like this Angiostigmata mite, which was recently discovered just outside of one of the oldest acarology units in North America. So the Ohio State University, and this is called the dragon, or the dragon mite, the Buckeye dragon mite, which are worm-like because they're in deep mineral soils and have to go through these tight interstitial areas. We see these different adaptations with the mite. So this is a species that we just recently discovered and described right here in Alberta, the cuculid, and they have these really thick front setae for predating on other soil organisms. Some are fast moving, like the Mesostigmata mites, which are predatory. Others, like this peacock mite, again, for defense, they're using this tail, this peacock tail that flicks over to defend against predators. And others have thick armor. So instead of being fast moving, they have or developed for finding predators. They're grazers of the soil community. So they're like the deer or moose within the soil, where they're feeding and requiring heavy armor to defend themselves against the predators. And scientists have studied these adaptive behaviors, and much has been learned about them, but it's very difficult because they're so tiny. They're usually between 150 microns to 2 millimeters in length. But still, scientists have figured out things about them, such as some of the ore-batted mites. They secrete or use cuticular hardening agents to turn themselves into little rocks so they can't be penetrated by other predators. Some use cyanide to fight against predators. They also have incredible sexual adaptations. So Dr. Heather Proctor, for example, found this water mite that the male uses the same vibrations as prey, so such as copepods, to get the female to move and towards it, and then it can present its spermatophores. There's a really funny cartoon on the oatmeal, which talks about how the red velvet mite creates these love gardens and tries to bring the females into its garden, and other males will come along and destroy the garden. So this is a really fun cartoon to look up. There's all sorts of ways. So because they're so connected to their habitat and they're so tiny, lots of different ways have come about to try to bring them out of that habitat so we can study them. One of the most popular is the Berlese, or the modified Tolgren funnel, which uses heat to dry out. And in this case, with the modified Tolgren, it's using light to dry out the soil and warm it up so that the soil organisms move down and drop into a cup. This is a collapsible funnels that can be used out in the field. Often this starts with first collecting the habitat to bring it back to these funnels. So using soil cores or trowels, or luring them or trying to bring them to the habitat using litter bags. Often washing is used. So here there's pails of sand being washed, and then the organic matter at the top is brought off to look through, often using hydrocarbon flotation to kind of collect the organic material to then look for those mites. Or if you're a bug hunter like me, you like to hunt for them, you can also just stare at rocks and stare at sand and try to pick them up with a paintbrush. And this is how we've actually found the cuculid mite in southern Alberta and gotten a better understanding of its range extent. Mites are diverse. They're also one of the least well-known groups. And so worldwide, currently there's about 63,000 described species from 528 families. But we're estimating that there's 500,000 to 1.5 million species globally. When you take that lowest estimate of 500,000, that's 12 to 13% have been actually described. Canada, we're at about 3,000 described species from 311 families. If you take the lowest estimate of 10,000, that's about 30% described. In Alberta, fresh off the press, we know that there's about 800 described species from 218 families. We haven't done an estimate yet, but we know that there's 1,376 records to date, which includes both undescribed species and a lot of records that are still at genus or family level. So we know that those numbers are still going up from what we currently know today. The Alberta Biodiversity Monitoring Institute, Sid gave a really nice intro to that, so I won't go into too much detail, but I wanted to point out that the mites that we study are from the main ecosystem health grid. So this is a grid of 1,656 sites across the province that are about 20, systematic as in they're about 20 kilometers apart across the province. And we're studying indicators at these, our study includes looking at indicators across these sites. And those indicators include using automatic recording units to look at birds, mammals, and amphibians, and examining collected specimens. So not everything can be identified to species using ARUs. So we also physically collect specimens and bring them back to the lab to identify them. This includes aquatic invertebrates, bryophytes, lichens, orobatid mites, and vascular plants. So you might ask, well, why orobatid mites? And so the main reason is that the orobatid mites really connect us to soil health. What is soil health? Well, it's a very large definition, but the definition is that it's the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And so soil has kind of three main components. There's the physical component, the amount of sand, silt, and clay in it. There's the nutrient component, or the chemical component, which includes those nutrients that sustain plant growth, but there's also a biological component. And so it's full of living organisms. And so the soil health needs to include that focus on biodiversity. And so the biodiversity in the soil can really be impacted by human footprint and management practices, and has been the main focus of ABMI. And soils are considered one of the most biologically diverse habitats on Earth. And it's been estimated that in 1 gram of soil, it can contain up to 1 billion bacteria cells, up to 200 meters of fungal hyphae, a wide range of organisms, including nematodes, earthworms, and arthropods, and comprising tens of thousands of species. That's just 1 gram. And 1 teaspoon can contain more organisms than there are humans in the world. So it's full of biodiversity. All of these organisms constitute a food web. So this is the underground food web, and they occupy different trophic levels, just like we have above ground. And this results in output of nutrients that allow for above ground plant growth. So studies focused on agricultural production have found that non-pest soil organisms increase plant growth by 46%. So they have a very important function within the soil and above ground. So orobatids are typically the most abundant soil arthropods in the soil. Typically at least 150,000 specimens per square meter of soil. It's been found up to 500,000 orobatid mites in forest soils per square meter of soil. And they occupy different trophic levels as they break down litter, and they also feed on fungi, bacteria, algae, nematodes, and other organisms in the soil. So one of the main reasons that ABMI has included orobatid mites as an indicator species is this role in soil formation and functioning. So they're providing us with ecosystem services in that their direct role is in decomposition, soil formation, and pest control. But indirectly, this turns into food production, soil conservation, carbon sequestration, water cycling, nutrient cycling, and pest and disease regulation. ABMI also uses orobatid mites because they fit indicator qualities. Because of their high diversity and that they're found everywhere and that they have such high abundance, there's a strong likelihood that we're going to get a signal from them. So it's not very often that we get an absence of orobatid mites in our samples. We usually get a signal. They are, unlike most arthropods, they're case-selected, which means that they have low reproductive rates and they live for multiple years. So this creates these stable populations that are really affected by environmental disturbance. They're easy to collect, so we have easy protocols that make it so that they can be sampled quickly in the field with a single visit using those soil collections and then identified in the lab. And we have the taxonomic resources to identify them. So this all makes it so that we can actually do the job. Another reason that ABMI uses them as an indicator is that they're used globally as an indicator. And so we've found in the literature that they've been used to look both at human footprint or those human related disturbances, as well as to look at habitat and environmental properties. And these, of course, overlap, such as change in pH may be connected to pollution. So how does ABMI go from data collection to data analysis? So first, we're going out to those sites. So there's the monitoring center for ABMI does our field work, and 1 hectare, each site has a 1 hectare area that's cut into 4 quadrants. And so at each of these, at the northeast, northwest, southeast, and southwest corner, about 10 meters out, there's a 1 meter plot set up where at least 4 soil cores are taken. And these are laid out and the mineral and organic soil are separated. And then each is mixed separately. And 500 mils of the organic and 250 milliliters of the mineral soil are put separately in bags and are sent back to us in coolers to the lab. If they cannot get the required amount, then they continue to sample until they can get 500 milliliters of soil. When it comes to the lab, the soil, we do all the logging to make sure we've got the right samples. And then we put the soil on modified Tolgren funnels. So there's this heat source at the top which then warms the soil up, and the critters move through and drop into a cup of 95% ethanol, so that we have the preserved also for DNA. And then they are sorted using a dissecting microscope. So we pick out all of the orbatid mites, and then some of them can be identified using a stereoscope to species level. Those that can't are slide mounted, and then we identify them using a compound microscope. So how is our data used? The first is much of the work so far has been looking at individual species, and I wanted to point out the biodiversity browser. And this is where you can find a lot of information on each species. So if you type in, for example, Atropocaris striculus, this is the pink box mite. So it's normally a pink color when it's not on an SEM. We have the range where we found it so far. So these little rust colored dots are where it's being found or collected. The lighter gray, the larger ones, are where we've collected but haven't found it. And then these smaller, tighter circles are where we haven't collected yet. We have information on species habitat association. So this is showing relative abundance. So it's the abundance of one orbatid mite species divided by the total abundance of all orbatid mite species in a site. And so we're finding that Atropocaris striculus is particularly associated with old stands of deciduous and mixed wood forests. And we have information on how they're being affected by different sectors. And so this is divided to agriculture, forestry, energy, rural, urban, and transportation. And what I wanted to point out particularly, first, the under footprint means that, so we have under footprint and regional. Under footprint means that this is how the species is affected specifically under agriculture versus how it's affected across the entire region. So the difference here is the pink box mite then, with specifically in lands that are agriculture, it's reduced by 45%. But because there isn't a lot of agriculture in the forested region, that decrease is quite minimal at 2.3%. So species, each individual species is responding differently to these sectors. But each species also differs in its response to these footprint types. So this is what we just saw with the pink box mite. The six-dimpled shelf mite, Tectocephalus saracensis, shows a completely different response. And so in agriculture, we actually find it's increasing by 522%, decreasing in forestry, which is interesting in comparison to these other areas. When we also have information on how their predicted reference conditions. So if we backfill the province with the natural habitat that was there before humans came along, for the pink box mite, it was at higher levels, particularly in the Parkland region, in comparison to the current conditions. And so again, we can see those differences between individual species, where we've seen a decrease over time with Atropocaris striculus, but we've seen an increase in time with Tectocephalus saracensis. So this is, we call these decreaser and increaser species, which then moves into intactness. So a lot of the work that's been done by ABMI to date has been to look at intactness of the province. And what this is it summarizes the current status of species. If we look at natural habitats that don't have any disturbance, we're going to find a high number of decreaser species and a low level of increaser species. Whereas we get into highly modified habitats, we find a low level of decreaser species and a high level of increasers. And so this turns into an intactness index, which can then be used to look at, again, intactness across the province. When we look at all species for all groups, so this is including the mites plus birds, mammals, vascular plants, bryophytes, and lichens, this is what the intactness looks like across the province. So low intactness is white and high intactness is blue. So again, where, as would probably be predicted, when we have high human, humans, we have low intactness. Where there isn't a lot of human footprint, we have a high intactness. There's also other metrics, and I'll just focus on the orobatid mites here. So we have that intactness for orobatid mites. There's also information on species richness, and what's kind of cool here is to see how their species richness, so the dark brown is 100%, so higher richness in these high elevation areas such as the Caribou Mountains and Birch Mountains. And then uniqueness is also quite interesting, where we're finding, I believe this is more the Shield area, and again, around those high elevation areas. Southern Alberta is also quite interesting for species uniqueness, where we're seeing that the military bases, which are quite high in natural habitat, have higher uniqueness along with the river valleys, and the farther extreme southern regions of the province are high in species uniqueness. Our data, when we talk about monitoring, it typically means that we're also going back to sites so that we can look at how we're seeing change over time. And this is trend. And we're getting to a point now where we're starting to be able to do that, to monitor trend, because we've been sampling. Some sites now have been sampled 2 to 3 times. So we can look at trend. Here's an example that came out, a report that came out last year on the ALPAC forest industry forest management agreement area. And so there's 154 sites within the area, and then there's an area of ecological interest that surrounds it. And so there's been kind of a comparison between the forest management area itself plus the surrounding. For the mites, what we found is that it's 93.2% intact on average, and this has declined from 94.3%. So between 2010 and 16, so over the 6-year period, and that it's higher, intactness is higher within the forest management agreement area in comparison to the area of ecological interest, which is at 89%. So 11 species associated with old deciduous and mixedwood forests were examined in this report. And so when we look at individual species, we find that all of them have decreased over that 6-year period. Some of them not very much, so just a 0.1%, whereas some of them is more significant. So Orbitella jacoti has decreased from 89.1 to 85.7%. And almost all species are still more than 90% intact. There's 3 that are at a point where they're less than 90% intact. We've also been using our data to look more at mite community analyses. So this is an example we've been working on over the past year within the oil sands region. So the oil sands region, these are our sites across that area. So 420 sites, and this is a nice mapping method on that ABMI has on their mapping portal to show the footprint, human disturbance types within that area. So we have mines, both active and abandoned well sites, seismic lines, pipelines and transmission lines, urban industrial, transportation, forest harvest, and cultivation. And so we've looked at these and compared them to natural undisturbed habitats, as well as looked at climate and space. And I don't want to get too bogged down, but what I wanted to show a visual showing that these are linear regression plots for orobatid mite total abundance and Shannon's diversity. And species richness was very similar to Shannon's diversity. So we looked at these compared to 21 land cover and climate space variables and found, so if we look at the lines here, as you go to the left, these are lower in abundance or diversity compared to as you go to the right. So for energy footprint, or in total, we're finding that cultivation and mines, well sites was also significant for the decrease in abundance compared to our natural habitats. It's interesting to point out that the soft linear is very similar to, and forest harvest is very similar to the natural vegetation. Diversity, it changes, and species richness changed a bit more. And so here is our natural vegetation. So forest harvest was very similar, but we're starting to see a decrease with the soft linear. So here's our seismic and our transmission lines, and then those more intense disturbance types, mines, well sites, urban industrial, roads and cultivation are decreased more in terms of the diversity of orobatid mites. We also did a redundancy analysis and found that there is a community composition difference. So we're finding species composition is different between upland and lowland types, as well as with those more intense disturbance types. So we're getting different species in these different areas. And we also use our data to increase our knowledge of mite taxonomy. So first, species detections and collections. So we're averaging over 6,000 specimens that we're identifying per year for ABMI. When we started, we knew of about 160 species in the province. So this was back in 2007. We've now detected over 287 species. So this includes morphospecies. And as some of the species we have not detected yet, but are known for the province, we now have 384 species detected in the province, including 223 new records to Alberta, 33 new to Canada, 9 new to North America, 15 described as new species, including these species, over 70 that may be new to science and yet to be described. And it's also, all of these have been curated and accessioned into the Royal Alberta Museum Invertebrate Zoology collection. So there's over 80,000 from ABMI, as well as probably tens of thousands more from our own research, other research projects. Our data is also used to refine species concepts. And this is kind of a fun example. We were calling these two species Adenozides rudentiger until they came in, both morphotypes came into our single sample. So this started to happen when we collected more in the mountains. So that's the area that they're found. So the question then becomes, okay, are they different species? And we've found at over 10 characters that differentiate them, including their color and their size. But then the question becomes, well, which one is the real Adenozides rudentiger and which one is a new species? And so going back into the literature, we found Adenozides rudentiger was originally collected by Marie Hammer back in 1948 at the Jasper railway station. She only collected a single female, and she identified it or described it as being dark brown and quite large, so over 500 micrometers in length. And so, but Valbehan Peltier in about 1986, she couldn't find the holotype. There was only a single specimen. And so she redescribed it based on the specimens that were collected in Western Canada, including some collected in Jasper, but noted that it was smaller than the original description. And so what we found is that this is the species that was redescribed as Adenozides rudentiger. This is likely, it's hard to tell because we don't have a holotype, but this is likely the original species. And so we now need to redescribe both and give this one a new name. So kind of a fun story. Our data is also used to develop publicly available resources. So we have all of our terrestrial field collection protocols are online. So this is very detailed information on all of our field collection methods, including not only our soil collection methods, but all collection methods for the 1 hectare plots. We have very detailed information on our standard operating procedures, including how we process the soil in the lab and do the taxonomy and curation. These are online at the ABMI website. We have a pictorial guide to identify orobatid mites to family, genus, and often to species level using the stereo microscope. This is not yet online. We're wanting to update some of the photos, but if you want to email me, it's available. I can send it to you. And we've been keeping up what we call living documents to provide record of Alberta's mite fauna with keys, species descriptions, locality, and ecological information. So this, the orobatida one is especially good for having full of those keys and descriptions, including the morphotypes, so that we can continue to name morphotypes with the same name. This just came out at the end of August, which has updated all our records for the Acari, including those 1,376 records that we're aware of in publications and unknown or unpublished records as well. Our data is also useful because it's at species level. It can be used for assessing conservation status. So looking at the general status of species across Canada. And so here I wanted to point out, because we've been monitoring those 13 years, we're getting to a point where we're similar to other areas where a lot of work has been done by acarologists. So BC, Ontario, and Quebec. There's still some gaps across the country. So Saskatchewan, actually it's quite interesting. 2 years ago, there was 15 records, but Monica Young produced a paper in 2019 that's increased this by, it's tripled it through that one single publication to 43. But seeing this discrepancy here, those 200 extra species did not just come from the mountains, because we're actually just starting to get into this region as well. There's a lot of habitat here that's very similar with the boreal, the shield, and the prairies, where I'm sure that the number of records is higher than that. So it'd be really interesting to collaborate with other people here to try to increase those records. Labrador, there's a lot of lichen and moss and habitat in Labrador. I'm pretty sure there's more than 7 species. Again, same as PEI with 8 species. There's work that can be done across the province. One thing that I didn't realize that with assessing conservation status is that we really need to have up-to-date records. So they should be collected within 20 years for us to be able to report on their occurrence within our region. And so there's some areas where we have not been to in the past, or collected in the past 20 years, at least in published records. So Northwest Territories, nothing's been collected in the past 20 years. This whole northern area, it would be wonderful to collect here and to be able to increase the records available for doing the general status. And some of these are, I would say, they're inflated because the number of species is likely higher. So because that recent publication came out, 74% of species are usable for looking at occurrence records, but but we think that there would be higher records. So lots of work that could be done, and it would be really fun to collaborate on this. We also have, this is mostly looked at the ABMI data, but we've also been collaborating with others. So we've been putting together a DNA reference collection for orobatid mites. So we have the CO1 for about 224 species in Alberta. We've been collaborating with acarologists across Canada to keep up those taxonomic records. Some nice collaborations with professors at MacEwan University, University of Alberta, to look at how mites are affected by invasive species. Again, some nice collaborations with University of Alberta and Alberta Environment to look at how they're affected, and as well as other soil mesofauna by reclamation methods, and then looking at natural disturbance, how they're affected by things like fire and or invasion of natural species such as aspen in the grasslands. So there's lots of we're always open to collaborations. The future opportunities. What is the passing grade? And so we're seeing decreases in or changes in mite species, but it would be really nice to get to a point where we're also looking at thresholds. So what is the right number, or the community that means that we have a healthy ecosystem? And it would be nice to begin to connect this information to the larger soil food web. So again, soil health is the chemical, physical, and biological components of the soil, and to look at those interactions and to get a better understanding of those thresholds for soil health. We know soil is going to change with climate change. There's going to be increased drought in the prairies, increased decomposition rates in the boreal. So looking at how mite communities and other soil biology is going to be affected by that. And then again, looking for those opportunities to collaborate with other jurisdictions to get a better understanding of both mites and soil in general, soil health. So with that, I'd like to thank you for your time. If anyone is interested in discussing this further or has ideas of how we could move forward with helping on using this in an applied way towards land management, we're definitely interested in discussing that. So please feel free to reach out.
Great presentation, Lisa. Thank you. I know I'm, always surprised to learn that something like a mite can be case selected. Just, to me, the two, I wouldn't have put them together.
Yeah.
So if anyone has any questions, I can pause the recording